This lecture is on Chapter 12, the cell cycle. The cell's life cycle is defined as the time from when it's first formed until it divides in two. With cell division, it generally results in genetically identical daughter cells. So this would be your somatic cells, your non-sex cells. So when we look at this cell division, you have the organization of the genetic material. The genome is just your cell's genetic information. It's a generic term for it. The chromosomes are the package form of DNA that we see for cell division. So this is specifically created for replication and distribution. Your somatic cells are all of the body's cells except the reproductive cells. Your reproductive cells, or gametes, are the sperm and egg. They're going to have half as many chromosomes as the somatic cells. That's important because they will join with another reproductive cell to have the full set of genetic information. We will talk about this type of cell division in Chapter 13. The chromatin is the complex of DNA and its associated proteins. This is the loose-coiled DNA. So you'll have chromosomes during cell division, chromatin in the non-dividing phase. So your DNA is made up of chromatin fibers when the cell is not dividing, and even as it duplicates. After duplication, it's going to condense into the chromosomes. Your sister chromatids are the two parts of the chromosomes. Each of them will contain an identical DNA molecule. The cohesins are an adhesive protein complex that's going to attach the two sister chromatids together. So sister chromatid cohesion refers to the attachment of the cohesion to the sister chromatids. The centromere is the specialized region of the chromosome where the two chromatids are most closely attached. So the cohesin will be what attaches them together. The centromere is the region where they are attached. So later on in cell division, the two sister chromatids separate and they'll be considered the individual chromosomes. So each nucleus is going to receive a collection of chromosomes that are identical to that of the parent cell. So in this diagram here, you can see you start out with your chromosome, the centromere, and then you will have chromosome duplication where you're going to replicate the DNA. It will condense and form a chromosome. And here you have the two sister chromatids. And then they will separate in cell division into two cells as two chromosomes. Mitosis refers to the division of the nucleus. And so this is only going to occur in eukaryotic cells. In prokaryotic cells, we don't have mitosis because we don't have a nucleus. We do have a similar process, though. Cytokinesis is going to be the division of the cytoplasm. This is usually going to follow the division of the nucleus. Exactly when this occurs will depend on the different types of cells. Meiosis is the sex cell division. This will give you non-identical daughter cells. These are only going to have half the chromosomes as the parent cell, so this would include your sperm and egg. During fertilization, you're going to fuse two gametes together, and this will resume the chromosomes back to the normal number. So your mitotic phase is going to alternate with interphase in a cell cycle. So in this diagram here, you can see interphase is the vast majority. It's about 90% of the cell cycle. And then we have the mitotic phase here, which is when the cell is actually dividing. So M phase is the mitotic phase right here. It's the shortest part. Interphase, 90%. During interphase, the cell is going to grow. It's going to copy its chromosomes. It's going to prepare for cell division. It's doing its normal job as the cell. We can divide interphase up into three phases. You have G1, S, and G2. In G1 phase, it's called G1 for the first gap. This is primarily cell growth. S phase is called that for DNA synthesis. This is where the chromosomes are duplicated and the cell grows some more. In G2 phase, this is the second gap. The cell grows some more and completes its preparation for going through M phase, or the mitotic phase. And then mitosis itself is broken down into 4 to 5 phases. It depends which book you are looking at. You've got prophase, prometaphase, metaphase, anaphase, and telophase. So telophase, for some reason, deleted off of your notes. So you're going to want to make sure you add this term into your notes. And I have it highlighted in green here where it will go. So your mitotic spindle, this is going to begin to form in the cytoplasm during prophase. It's made up of microtubules and proteins, and it's going to elongate by incorporating more tubulin subunits. And then it will shorten by losing those subunits. The centrosome helps to organize the cell's microtubules. It starts the assembly of the spindle. You have a pair of centrioles that are located at the center of the centrosome. The centrosome is not essential for cell division because it's not present in plant cells, but it is seen in animal cells. During interphase, a single centrosome replicates to make 2 centrosomes that remain near the nucleus, and then they move apart in prophase and prometaphase to be at the opposite ends of the cells by the end of prometaphase. So here you can see the centrosomes, the pair of them, that are going to move apart. So the aster is a radial array of short microtubules that extend from the centrosome. You can see them up here at the top of this cell. So the kinetochore are proteins that are associated with specific sections of the chromosome DNA at the centrosome. They face in opposite directions. And during prometaphase, some of the spindle microtubules will attach to the kinetochores when the kinetochore is captured by the microtubules, and the chromosomes will move towards the poles. The metaphase plate, this is in the plane that's midway between the 2 spindle poles. Here you can see where you would have your metaphase plate. This is where you're going to have the duplicated chromosomes line up during metaphase. Anaphase begins when the cohesins that are holding the sister chromatids of each chromosome are cleaved by an enzyme and will become separate. They will end up being separate chromosomes. So your non-kinetochore microtubules are going to be responsible for elongating the whole cell in anaphase. You have non-kinetochore microtubules from opposite poles of the cells that overlap, and it's more extensive in the metaphase region, and overlap is reduced in anaphase, is the motor proteins walk away from each other. So the kinetochore microtubules will have chromosomes attached. The non-kinetochore microtubules do not have chromosomes attached, and their primary job is to stretch out the cell. During telophase, you're going to have the nuclei reform. Cytokinesis generally begins in anaphase or telophase. Again, the exact timing of that is going to depend on the cell type. So when we look at G2 of interphase, here your nuclear envelope is going to have 1 or more nuclei in it. Your centrosomes form, and chromosomes will form during the S phase, but they have not yet condensed. So here in this picture we can see G2 phase, you can see the chromatin. It's duplicated here, but it's not actually condensed into chromosomes. You've got your centrosome pairs, your nucleolus, the nuclear envelope, your plasma membrane. So during prophase, your chromatin is going to form chromosomes. Your nucleoli disappears, the sister chromatids form, and the mitotic spindle forms. Here you can see where the chromosomes have condensed. Here your centromeres are going to begin to separate. In prometaphase, your nuclear envelope is going to begin to fragment. Your microtubules are going to extend, and the chromosomes become more condensed. Some of the microtubules will attach to the kinetochores and pull apart, and you'll have the non-kinetochore microtubules interact. So metaphase is the longest stage. The centrosome is at the opposite poles, and it convenes on the metaphase plate. Your chromosomes will all be on the metaphase plate, and the kinetochores of each sister chromatid attaches to the kinetochore microtubules from the opposite pole. In this picture here, what is really distinct is seeing all of those chromosomes aligned on the metaphase plate. So in anaphase is when you're going to have those sister chromatids are going to separate. It's the shortest phase. The cohesin protein cleaves, which allows the sister chromatids to separate into chromosomes. The chromosomes are going to move towards the opposite ends, and the kinetochore microtubules will shorten and the cell elongates. So here you can see this happening. It's pulling apart and separating. Here you have telophase and cytokinesis. You can see you're forming a new nucleolus. The nuclear envelope begins to reform. You have the cleavage furrow forming where it's starting to pinch off. And you'll end up with two new cells. So in telophase, you end up with two new daughter nuclei, a new nuclear envelope. The chromosomes will become less condensed and go back to chromatin, and you have two new cells. This diagram here is showing where you've got the microtubule, and as the chromosome moves, here it is moving and breaking off the tubulin subunits. So it kind of builds the microtubule by putting the subunits, the pieces on like Legos, and then it breaks it down by pulling the Lego pieces off. So cytokinesis is division of the cytoplasm. It's usually well underway by late telophase. You're going to form the cleavage furrow in animal cells, which will allow it to pinch in two. So here is the cleavage furrow. It gives the appearance of a shallow groove in the cell surface. This is where the old metaphase plate was. On the cytoplasmic side of the furrow, you actually have a contractile ring of actin filaments that interacts with myosin and causes the ring to contract. It's similar to having like a drawstring in the waist of your pants, that when you pull the drawstring, it starts to shrink and the pants come in around the drawstring to pinch into two. In plant cells, you don't have a cleavage furrow because of having cell walls, so instead you'll have a cell plate. The vesicles from the Golgi move along the microtubules to the center and coalesce to produce this cell plate that will create a division in the two cells. So you can see the difference here, the cleavage furrow with the contractile ring versus the cell plate being deposited. So in prokaryotes, you're going to do binary fission as a form of asexual reproduction. These are single-celled organisms. You cannot do mitosis because they do not have nuclei. So your bacteria and archaea will be in this category. And they do have a single circular DNA. So what will happen is you will copy that circular DNA, you make a copy of the chromosome, and you end up with two copies. You do this by starting to split it apart, where you're going to have these tiny little origins of replication. And as it continues to copy, they're going to pull apart further and further. The origins become further apart. Eventually, you have 2 copies of the DNA, and you will be able to form a division between the 2 cells, and you end up with 2 new daughter cells. So the origin of replication is the process of cell division that begins in separating the 2 circular chromosomes. It will produce 2 origins while the chromosome replicates, and the cell is going to elongate to about twice its normal size. The plasma membrane will grow inward to create the division into 2 daughter cells. So mitosis and binary fission are not the only 2 plans out there for cells to be able to divide. In the evolution of mitosis, it appears that it had the origins in the prokaryotic method of binary fission. Some of the proteins involved in prokaryotic binary fission are related to the eukaryotic mitosis proteins. We think there is possible intermediate stages in evolution that may be seen in the dinoflagellates and diatoms or yeasts. So the dinoflagellates, their replicated chromosome attaches to the nuclear envelope, and it separates as the nucleus elongates prior to dividing. So here you can see it being attached, the chromosome being attached and that pulling it apart. The diatoms and the yeast, they have a spindle within the nucleus that separates the chromosomes. So you can see this in here stretching across. So when we look at the eukaryotic cell cycle and molecular control, the timing and rate of cell division really does vary with the different types of cells. Some cells very rarely divide, others divide fairly frequently. So we have pretty good evidence for there being cytoplasmic signals to tell the cell when to divide. The cell cycle is driven by specific signaling molecules in the cytoplasm. This was tested by fusing cells in different phases of the cell cycle. And when that happened, you actually pushed the other cell into that phase. It went into whatever phase it was receiving from the cell signaling molecules that were in its environment. So here, when you had cells that were in the S phase and G1 phase, you ended up with them both going into the S phase because it picked up the signals. Here you had the G1 phase and the M phase. Both cells ended up going into the M phase because of the cytoplasmic signals. So the cell cycle control system, you have a cyclically operating set of molecules in a cell that triggers and coordinates the events in the cycle. We have checkpoints as control points along the way in the cell cycle, where it's going to receive stop and go signals that will help to regulate the cycle. In animal cells, they generally have built-in stop signals, so by default it is a stop signal and there has to be a message for it to be overridden. A lot of the checkpoints actually come from surveillance mechanisms inside the cell. So when we look at the cell cycle here, we have a G1 checkpoint. This is a restriction point in most mammal cells. If you get a go-ahead to progress through G1, you're usually going to go ahead and progress through G1, S phase, G2, and M phases. If not, here is where you would take a detour and go into the GO phase. The GO phase is a non-dividing state of cells. Most cells in the human body are actually in the GO phase. Your mature nerve cells never divide. So occasionally some of these cells can be called back from the GO phase with external signals and clues. But for the most part, these are considered your non-dividing cells. So we have a cell cycle clock that's going to help to control things. It's going to use the protein kinases So these are enzymes that are going to activate and inactivate other proteins by phosphorylation. So these can be present at constant concentrations in a growing cell, but in an inactive form. So a cyclin is a protein that attaches to a kinase to activate it. And a CDK or cyclin-dependent kinase is a kinase that requires a cyclin. So these levels will actively rise and fall with the concentrations of the cyclin partner. MPF, or maturation promoting factor, you can think of this as your M phase promoting factor, is going to be controlled by these cyclins. So here over on the left you can see the cyclic nature of this, where you're going to have the cyclin concentration build up, and once the cyclin concentration peaks, you're going to have the MPF activity, and that's when you're going to have the M phase. So you can see again it builds up here. At the peak you have the M phase. It builds up again, the peak you have the M phase. So cyclin synthesis begins in the late S phase, so right here is where you're going to start to begin to synthesize those cyclins. And this will happen through G2, and it gets protected from degradation and starts to accumulate. Then the cyclin is going to combine with the recycled CDK or cyclin-dependent kinase, and this will produce enough of the MPF for the cells to get past the G2 checkpoint. The MPF is going to promote mitosis by phosphorylating various proteins, and it will reach its peak during metaphase, so here during mitosis. The cyclin component of the MPF degrades during anaphase, so it's going to start to break this down, and this will terminate M phase and it will enter into G1. And then in G1 your cyclin degrades and the CDK is recycled. So your MPF is going to act directly as a kinase and indirectly activating other kinases. This causes phosphorylation of various protein and nuclear lamina to fragment the envelope. This contributes to the chromosome condensation and the spindle formation in prophase. So during anaphase, it initiates the process to destruct its own cyclin to switch itself off. So in animal cells, it appears that we have at least 3 different CDK proteins and several different cyclins at the G1 checkpoint. So you can think of this as being more of an example of how this process works, but there are multiple different types of CDK proteins. So we do have other stop-and-go signals, your internal and external signals at checkpoints. The internal signal at M phase, during anaphase you have separation of the sister chromatids. It does not begin until all are attached at the metaphase plate. This is really important because only when the kinetochores are all attached and the regulatory protein is activated, this will ensure that you have them all lined up so they can all split and you don't end up with extra or missing chromosomes on one side. Growth factor is a protein released by certain cells that's going to stimulate other cells to divide. So far, we've discovered more than 50 growth factors. One example is PDGF, the platelet-derived growth factor. It's made by the platelets. This is required for the division of the fibroblasts. So this gets released from injury, and then you'll have proliferation of the fibroblasts to help with the wound healing. So another thing that will control whether or not cells are dividing is density-dependent inhibition. This is an external signal when cells become too crowded to stop dividing. Another important regulatory mechanism is anchorage dependence. Most animal cells need to be attached to a substrate to divide. So if they don't have a home to attach to, they won't divide. So loss of cell cycle control is seen in a lot of cancer cells. And so most cancers are not going to be just the result of one thing going wrong. There can be multiple things that have gone wrong in these cells. So the idea that you would have one thing that's going to fix all cancers is extremely unlikely. So when you look at cancer cells, they don't follow the normal signals regulating the cell cycle. One of the things, they lack density-dependent inhibition. So if you have a tumor that's growing, it doesn't care that it's crowding out other tissues. One of the ways that tumors do cause problems is they will grow and actually start to smash and compromise other tissues, taking their nutrients. Another problem you can have is anchorage dependence. So this is a problem when cancer cells begin to metastasize. Normally, a good healthy cell needs to be attached to something before it's going to divide. With a cancer cell, it will say, you know what, I don't really care that I'm not attached to something, and they will divide anyway, and this will allow them to spread into other tissues. So most cells are going to stop dividing when their growth factors are depleted. Not so much with cancer cells. If and when they stop dividing, they seem to do so at random points. They can divide indefinitely with a continual supply of nutrients. So one of the things we know with most normal mammal cells, in a cell culture they can divide between 20 and 50 times before they kind of age off, they stop dividing and they die. With cancer cells, that's not the case. This was actually one of the reasons why the HeLa cells were so important. Years ago, they thought that no cells would be immortal, that it was just part of who they were. Cells would divide up to about 50 times and then they would divide. And there was a researcher who was convinced that there would be some immortal cells, so he collected cell cultures from several patients. So at the time, there really wasn't any laws or rules about what to do when you would collect cultures from patients, and nobody really believed that he would actually find these immortal cells. So as he's doing this, he at one point collects these cervical cancer cells from this woman Henrietta Lacks in 1951. And to everyone else's surprise, these cells just kept on dividing in cultures. So he was very interested in research, so he ended up sharing these cells with other researchers. And eventually, they were being shared with more and more people, and there was a need for them, and they were taken over and produced commercially. So the way that they are named, HeLa cells, is from the first two letters of her first name, the last two letters of her last name. So there's actually a book that talks about what happened in her scenario, because the family was never compensated at all for this, and they didn't really understand what was going on. They were kind of confused by the fact that there were these cells from this woman still out there in cultures. So transformation is the process that converts a normal cell to a cancer cell. This actually occurs more frequently than we have cancers, but normally your immune cells are going to destroy it. So if it evades destruction and proliferates, it can form a tumor that can progress to being cancer. So a tumor is just a mass of abnormal cells within otherwise normal tissue. A benign tumor is those abnormal cells when they remain at the original site. Most of these can be completely removed by surgery. And depending on where they are and how those cells are behaving, sometimes they're not actually removed. One of the things, they can be problematic just because they don't really care that there's not space for them to grow, and they'll continue to grow and compromise the other tissues. With a malignant tumor, it becomes invasive enough to impair function of one or more organs, and this is when it's considered cancer. So you can have a tumor grows from a single cancer cell. It can start to invade the neighboring tissue, and then it can start to spread through the lymph and the blood vessels to other body parts. So that's when you have the metastatic tumor. So when they start to form another tumor, that is your secondary tumor or your metastatic tumor. So malignant tumors have cells that are abnormal in many ways beyond excessive proliferation. A lot of times they'll have unusual numbers of chromosomes. Their metabolism may be disabled or provide no useful function to the organism. They can change to their surface so that they no longer need the attachments, which allows them to spread. They can actually signal molecules for angiogenesis. So when they take off and find a new home, they will actually send out signals for the body to build them a blood supply and deliver nutrients to them. So with metastasis, you have a separate tumor from the original tumor, and it's proliferating in a distant location. So with cancer treatments, sometimes a tumor can be removed surgically, other times it can't. High energy radiation can be used to treat localized tumors. These will damage the DNA in the cancer cells, and it will damage the cancer cell DNA more than the normal cells. The majority of the cancer cells lose their ability to repair damage, so the idea is that you can damage those cancer cells enough to actually kill the tumor. This is useful when you have a localized tumor. You can't do radiation all over the body because it's too damaging. Chemotherapy uses drugs that are toxic to actively dividing cells, and it spreads through the circulatory system. This is used to treat either known or suspected metastasis. So this will diffuse throughout the body, hoping to get any of those cells that may have spread around. Taxol is a drug. It was actually developed in this area here that came from the yew tree, and it freezes the mitotic spindle by preventing the microtubule depolymerization. So it stops cells from going past metaphase. So it can be used in cancer treatments to try and stop and decrease cell division. So generally, cancer drugs do have a lot of side effects, and this is due to the drug effects on the normal cells. The hope is that you will damage enough of the cancer cells to kill the cancer before causing too much damage to the normal cells to kill the whole individual. Sometimes that works, sometimes it does not. So there are a couple of things that are interesting that if you're looking for more information, there is a Nova video called Cancer Warrior. Sometimes you can find it on PBS or the Nova apps or even on demand. I also found it on YouTube, so that's a good place to look for it as well. That talks about angiogenesis inhibitors and how they were developed. If you're interested in more on how these HeLa cells came about, there is a book, The Immortal Life of Henrietta Lacks, that goes into that story by Rebecca Scoot. I know that the Pierce County Library System had it. I read it and checked it out online that way. It's also available at normal places where you buy books. That is the end of chapter 12.